2D-GEL electrophoresis. 2D phase is a technique called two-dimensional gel electrophoresis used to separate and analyze complex protein mixtures. It separates proteins in two dimensions. First, by their isoelectric pH in an isoelectric focusing step, and then by their molecular weight in a second step using SCS phase. This process can separate thousands of proteins simultaneously, which are then visualized as spots on a gel. It was described in 1975 by P.H.O. Farrell. It is used for separation and fractionation of complex protein mixtures from biological samples. 2D phase separates proteins depending on two different steps. In the first dimension, proteins are separated according to their isoelectric point, that is pi. The second dimension is STS polyacrylamide gel electrophoresis, that is STS phase, which separates proteins based on the molecular weights, relatively molecular weight. Principle: 2D-GEL electrophoresis separates proteins based on their isoelectric point and molecular weight. The first dimension uses isoelectric focusing to separate proteins by their intrinsic charge pi, with proteins migrating to a position where the pH equals their pi. The second dimension uses STS phase to separate the proteins from the first dimension by their molecular weight. First dimension, isoelectric focusing, proteins are separated based on their isoelectric point, which is the pH at which a protein has no net electrical charge. The proteins are loaded onto a gel strip with the pre-established pH gradient. An electric field is applied, causing proteins to move until they reach the pH of the gel that matches their pi, at which point they stop migrating. Second dimension, STS. The proteins from the first dimension are transferred to a second gel. Sodium dodicylsulfate is used to denature the proteins and coat them with uniform negative charge, so they are separated solely by size. An electric field is applied to the gel and the proteins migrate through a gel matrix based on their molecular weight, separating them into distinct spots on the gel. This process results in a two-dimensional map where each spot represents a unique protein or a protein variant, with its position indicating both its pi and molecular weight. Procedure: sample preparation. The native samples need to be converted to a physicochemical state suitable for the first dimension of isoelectric focusing and keep the native charge and molecular weight of the constituent protein. Isoelectric focusing, the first dimension, proteins are separated according to their isoelectric point, that is pi, the pH at which the protein negative charge is 0, in a process known as isoelectric focusing. STS phase, that's second dimension. STS polyacrylamide gel electrophoresis, which separates proteins based on their molecular weight. Visualization of protein. There are various methods for visualization of proteins, but the most commonly used are Q-massive blue staining and silver staining. Applications: systematic separation, quantitation, and identification of many proteins from a single sample. can separate thousands of proteins simultaneously, can detect post-translational and co-translational modifications which cannot be predicted from the genome sequence. Proteome analysis, cell differentiation, detection of disease markers, drug discovery, therapy monitoring, cancer research, purity checks, microscale protein purifications. Thank you.